Hey, what's going on, everybody? Um, Today, I just want to do a quick video lecture on 6.3b, immunity, and this will be part of part one. So make sure you look at the three parts for this week and complete um, those and submit those. But I want to quickly go over the slides with you guys and talk about immunity, which is actually really interesting and lifelong lesson, I guess. So that way, throughout life, you know what is happening. Let me quickly screen share with you guys my slides. Okay. So as you guys can see, we are on 6.3b. I know I gave you guys a few weeks to catch up, so that's kind of the last few weeks. But um, the first thing I do want to talk about in this section is the white blood cells. You also can, the actual scientific name for white blood cells are leukocytes. And they're basically responsible for first identifying the pathogens, and number two, protecting the body from the infection. So two main missions of white blood cells is identify and basically destroy, identify and protect, whatever that protect might mean. A lot of times it is destroy. So search and destroy type of thing, kind of like a military force for our body to protect us from pathogens that are going to come in and might cause harm. Okay, so a lot of times you'll see the word leukocytes. That just means white blood cell. Make sure you kind of note that. And there's a few different types. There's just three here: eosinophils, monocyte, and lymphocytes. You might have heard of the lymphatic system. A lot of lymphocytes are found there. So that's kind of what you see. I want to okay, so phagocytes specifically, one type of a cell is called a phagocyte. They're a type of white blood cell that is able to move out of the blood. So make sure you understand that white blood cells flow through the blood. They're just like red blood cells. Everything is that's in the blood. White blood cells travel in the blood. As they travel in the blood, the specific white blood cell called the phagocyte, it can actually move out of the capillaries because remember, capillaries are super small and they have like little pores where gas exchange can happen. These phagocytes are so small they can actually get out of the capillaries and go to the site of the infection. So when you have an infection or some kind of a damage to where there's bacteria or other things that came into your body, let's say you got cut and whatever was on the surface that cut you had some pathogens on it, that site or the infection or the damage, usually there's like a chemical message that attracts these phagocytes to leave the capillaries and go to that site of, or kind of like a maybe like a call, but a chemical call. Okay, it's like, hey, there's injury here, hey, there's an infection here. That's when phagocytes actually go into or out of the capillaries and into the site of the infection. When pathogens are detected by these phagocytes then engulf and digest them with enzymes. So engulf meaning kind of like they swallow them up. So if you see here, they kind of like open themselves. This is a yeast cell, so a pathogen, and they open themselves up and they technically kind of like gobble them up or swallow them up. That's what it means by engulf. Okay, they will engulf it or swallow up, and then they will release a bunch of really, really nasty enzymes to kill this pathogen that now is inside of them. So they will engulf it and release a bunch of enzymes to kill that pathogen. That's how phagocytes work, which is really cool. There's a lot on phagocytes, but that's just like the quick version. So phagocytes, non-specifically, are going to engulf pathogens via endocytosis. So remember that word is endo means inside, cytosis cell. So the engulfing process happens via endocytosis. And remember, endocytosis and exocytosis require energy, so there is some energy involved in that. But this process, when a phagocyte engulfs or takes in the pathogens into itself, we call that a phagocytosis. So we just combine two words, phago from phagocyte, cytosis cell. So phagocytosis is a phagocyte swallowing or engulfing something into itself and then to destroy it. So there's steps here. This is a really cool picture. You have a microbe or particle via chemotaxis. So there's steps here that you guys see to your right, 1 through 7. So if you see 1 through 7 on the right side, make sure you guys pay attention to that, because that these right here, these 7 will come back to be in like your review questions and assignments. So make sure you kind of read through that. But the picture here does a great job of telling us that we have a microbe. Oops, sorry about that. And it is going to travel. The phagocyte will open itself or kind of expand its membrane to make like a engulfing or a little well, so that phagocyte will go in there. Then ingestion, that's number 2, that's here, ingestion of microbe by a phagocyte. Number 3, the next thing that happens is you have a formation of phagosome. A phagosome is basically like a bubble where the phagocyte is trapped into, think of this kind of like a jail cell. So it's trapped in this phagosome. The fusion of phagosome with lysosome. So then what the phagocyte will do is you have a bubble of lysosome, which is the lysosome. And then the lysosome that has these little blue dots are going to be your enzymes. So digestion happens when these enzymes, this lysosome and this phagosome, they come together. They the lysosome will release these enzymes into the phagosome, and then that phagocyte is going to be then broken down. Number 5, digestion. Okay. Number 6 is formation of residual body, basically just garbage or the trash that's left over, because now it's all destroyed, it's broken down, it's ripped apart. So technically think of it like a chemical blow up. It just blows it all up, destroys it completely. And now this here needs to get rid of it. And that's what number 7 is. It's just exocytosis now. When this is exiting, it's going to discharge the waste. So this is a cool process. Make sure you kind of go through these steps when that question comes up. We also have pathogens that are going to enter the body. When they enter the body, they do initiate an immune response. So when a pathogen enters the body, is detected, there's a certain immune response that your body kind of triggers. Okay, what happens is if you see the antigen, okay, antigen is going to be that little popcorn looking yellow cloud thingy right here. Okay, and then your antibodies are going to be these Y-shaped green and purple structures. So proteins and molecules on the pathogen. So antigen here is the same word for pathogen. Okay, the antigen has there's proteins on there on the surface. Those proteins are going to be called your antigens. So that's going to be like, think of it like there's a bunch of flags or little markers, protein markers that the pathogen has on its outside, and that's going to be on the outside of that pathogen. That's where we'll see it. And then that's going to be on the outside here, your proteins. Okay, so the pathogens, this whole thing, the outside layer will have those, surface layer will have those antigens or protein markers or little flags like, hey, I'm not from this body, I'm foreign. Okay, when your body recognizes that there's these antigens on these pathogens, the body will then send antibodies. They are recognized as foreign bodies, right? These antigens, because they're not from your body. This is something that's from outside of you that came in somehow, either through a wound or something that you ate, whatever it is, right? It entered your body. So the response to these antigens being detected in your body, your white blood cells produce a specific antibody that binds to the antigens on the pathogen. So this antibody, this Y-shaped, right? This antibody will attach itself to the antigens of the pathogen. So this whole thing is a pathogen. Let's review that. The antigens are going to be on the outside. So the antibody will attach to the antigens on your pathogen. Okay. So when that happens, your lymphocytes are going to be those white blood cells. That's a different type of a white blood cell. So we talked about phagocytes, now we have lymphocytes. So lymphocytes are the white blood cells that produce these antibodies. So each antibody, okay, binds to a specific antigen. So it's a 1-to-1 ratio. So for every single antigen or every single foreign pathogen that enters your body, there's going to be one specific antigen. It's a 1-to-1 ratio. So you cannot have two different antibodies binding to the same pathogen. So it will always be the specific antigen that binds to a specific pathogen or antibody binds to a specific pathogen. Okay, so each antibody has a region that is very specific to the antigen. So when the antibody binds to the antigen on a pathogen, okay, so the antigens are going to be, remember, those are those little flags on the pathogen, those are those proteins. So antibody will attach to specific antigens, then it aids in controlling the infection. Okay, we're going to talk about that. So this is your antibody right here, it's a Y shape. And then if you look here, you have an antigen binding site. This is where the antigen as from the pathogen will bind. And if you look at it, they're very specific. So they're just trying to show you the cutout, right? It's very specific. So every antibody will be specific to one type of antigen. So that means for every sickness or for every, let's say, viral infection, bacterial infection, that specific antibody will only work for that specific pathogen, because the antigens are specifically different from one sickness to, from one pathogen to the other. So it's going to be always very specific. Okay, big deal there. So if you look at these diagrams, is a really good way to show you how antibiotics act. So antibiotics technically are going to destroy bacteria or pathogens that enter your body, mostly in this case bacteria because it's antibiotics. But so phagocytosis, so some antibiotics enhance phagocytosis, so they help bring that pathogen to the macrophage or to this white blood cell, and then some will help those pathogens pop. So they will literally, if you look here, they will literally come and cut their membrane and make them pop. Okay, so the presence of antigens stimulates the corresponding lymphocyte to produce large amounts, large clones called plasma cells. After time, enough cells are present to combat the infection. So basically, all that's saying is, when antigens are there, so you had a pathogen that entered your body, they have antigens on the pathogen. Okay, that lymphocyte will then detect, oh, foreign body. When it detects it, it produces a huge number of clones of these antibodies. We call them plasma cells, okay? A huge number of them. And then after some time, there's enough of those cells present, okay? Or enough of those plasma cells that produce all these antibodies present to go ahead and start the infection, killing process of the infection, okay? So combating the infection. After the infection has been dealt with, so let's say your body detects it, your antibodies go in there, your plasma cells create a bunch of antibodies, they go in, they do its job, they destroy it, right? And the antigens are no longer present. That's not the end of the story. So your body doesn't just destroy it and then calls it a day. So your body actually does something really cool, and what happens is the lymphocytes gradually begin to die. Okay, so if you guys see that underline there, so even though there's no antigens, the lymphocytes begin to die because there's nothing to fight. But that's not it. Okay, that's not the story. That's not the end of it, at least. So the story keeps going because some of these lymphocytes, they actually live on as memory cells. So that's why we say for every specific virus or bacteria, whatever your body fights and beats it, technically, okay, you can never be sick from that same infection because you create these memory cells. Okay, these memory cells, they stay inactive until, so they're there in your body, they're floating in your blood just in case this same pathogen, let's say, enters your body 10 years from now, 20 years, whatever that is. If it ever comes back with those specific antigens, now, so it's inactive, and then until the pathogen enters and this the immune system encounters this again, then you have this memory cells that have these already ready to go. So it's like soldiers specifically ready to go for this specific pathogen if it ever comes into your body again. So that's why they're called memory. It remembers this is it, and they stay in your body. They're inactive, but as soon as this same pathogen comes in, they quickly reproduce, And as a result, they confer an immunity to the pathogen and actually kills it so fast that you technically never feel sick from that same pathogen twice in your life, because you have these memory cells that your immune system kept for years and years and years, right, throughout your lifetime. Okay, so immune response. Let's quickly talk about that because you'll need that. Immune response. Okay, so you have this diagram here, kind of summarize the response of the immune system. Just so you know, you have antigens. So you have a bacteria in this case. It has these little flags on the outside. That's going to be your antigens. This here is going to be your let's just call it your white blood cell. They have all these specific names: helper T cell, blah blah blah. This is your white blood cell. It engulfs it. Remember phagocytosis, so phagocyte. And then this detection here that there's an antigen that tells the immune system to activate helper B cells, all these things. The end point of this is you have clones of plasma cells. Those clones then release a bunch of antibodies to combat it, but you also then have these clone of B cells that will then become your memory cells, okay? So now you guys are here at the review. Go ahead and you will see in this is part 1 of the 3 for this week. So for part 1, I created a Google Doc for these questions to be answered on there. So go into Google Classroom, and after you have watched this short lecture, you're going to jump into these questions and complete these. Please look out for the command terms: outline, describe, discuss. Make sure you answer them completely to these IB term, biology requirements, whatever these term means, right? So make sure you're summarizing. Describing is going to be obviously a long one. Discussing is going to be a long one, but go ahead and complete that, and then this will be the end of part 1 once you watch this lecture and answer these questions. And there's part 2 and 3. So go ahead and get that done and turn that in. All right. So be safe, and I guess kind of close to your graduation. So be safe, be good. Have a good one. Be safe.